This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 175, recorded on March 16th, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
How's it going out there in the West?
Oh, going well. Or at least work's going well. The weather's crummy. But
it was warm this week, right?
Yeah, it was warm. Now it's raining.
Your crocuses must be out too, right?
Maybe. What do they look like?
What do they look like? They're short, about 2 or 3 inches high, very colorful, purple, whitish, red.
Okay.
They're usually the first ones to come out.
Yeah, I don't think we have any on our property, but I'm sure we have neighbors who have those.
They're out.
Yeah.
Also joining us today, back again from microbiology and immunology at the University of Maryland in Baltimore, Matt Frieman.
Good to talk to you guys. How are you?
Yeah, we're okay. Are you you're in short weather down there already, right?
It's nice. It's been in the up 70s, a little over 80 a couple of days this week, so It's been beautiful.
Is that unseasonable?
Oh yeah, I think it should be in around 60 at this time of the year. So we've had obviously, like everyone else, had a very mild winter on the East Coast.
Yeah.
Cool. Well, Rich Condit is out in the western U.S. skiing. He just sent us a photo by email of him on the slopes, so he won't be joining us today. And who knows where Dixon is, of course. Anyway, we have a couple of cool stories to tell you about today. The first one, this is actually a story I have been meaning to do for a long time. And this isn't the paper, but it's a follow-up to the original paper I wanted to do. It came out in the Journal of Clinical Investigation. It's called Herpes simplex encephalitis in children with autosomal recessive and dominant TWiV deficiency. So we'll explain all of that to you. There are really a lot of authors on this paper. It comes from the PI, or the lead author, is Jean-Laurent Casanova over at the Rockefeller University here in New York. But it is a big collaboration, including people in France, Saudi Arabia, Tehran, which is, I don't know how, I haven't seen a paper with anyone on it from Tehran, Japan. It's really an interesting collaboration, and this is because of the patients that they're enrolling in this study.
Right.
This is a cool thing because I think, here's the general problem is you have a virus infection, it infects a lot of people, like the herpes viruses typically infect a lot of people, but most of them don't get sick. But then there are some who do get sick. And why is that? And most people think that it's some mutation in those individuals, maybe in their immune system, and that makes them susceptible. So here, the infection is herpes simplex encephalitis. So as everybody knows, just about everyone gets infected at birth or shortly afterwards with herpes simplex viruses. In fact, all the herpes viruses we all end up getting pretty much. And most people just have an inapparent or mild illness. And then, of course, in the case of herpes simplex, the virus becomes latent in your peripheral ganglia, and then periodically it gets activated, you get fever sores. But apparently, a number of the kids who get primary infections, so between 6 months and 3 years of age, they get herpes encephalitis. So that's bad because the virus goes in your brain, and it multiplies there, and that can cause problems. Now, this is a pretty rare disease, 1 in 250,000 individuals a year, but before drug treatment, it was bad. It was fatal, 70% mortality rate. With the use of acyclovir, you can control that, but apparently the kids have neurological deficits afterwards, so this is not something that you want to have. So in this paper, they asked the question, why do some kids get herpes simplex encephalitis? Obviously,
and this is something this group's been working on for a little while.
Right, because they had previously published. So what they do basically is they sequence the genome of these kids and say, is there anything obvious? And they previously showed that these kids have specific defects in parts of the innate immune system. The sensors in particular, the toll-like receptors that sense foreign molecules and turn on interferon and other cytokine production, they have defects in those. And this is a continuation of that. They actually find different mutations here, but it's a nice paper to go through to illustrate how this works. So I've always liked this story. I think it's really interesting. And I think most diseases where you have rare disease, I'll bet there's some kind of mutation like this. Not flu, for example, where almost everyone gets sick or a large fraction of people get sick. But one I was thinking of is vaccine-associated polio, where you give the vaccine, the Sabin vaccine, to 1 million kids, and one gets polio. There's nothing wrong with the vaccine. There must be some mutation in that child.
Well, another possibility is that there's a co-infection. I mean, that's something I've always thought of as a possibility. I mean, in this case, we're going to talk about genetic issues, but it's also possible that you have one virus going in that would normally be harmless, but on top of that, there's something else going on.
It's interesting that you should mention that, because just this week, a reporter sent me an article in a like totally non-scientific source. I don't remember what it was, but it was suggesting that often 2 or more pathogens are involved, but we only look for one because we do reductionist stuff. Like Koch made it really important to make pure cultures, right? So you focus on one. So Matt, you would be interested in this. This guy focused on the SARS outbreak as saying we tried very hard to get pure cultures and we found one virus, but it biases against multiple etiologies.
Yeah,
sure.
Of course, in the case of SARS, there's clear evidence that virus causes SARS, right?
Yes.
Right, exactly.
So, but he's right in that.
Poor choice of example, but in order to find an example, you'd have to find a situation where we didn't do that, but of course that's the procedure we use.
Well, when you culture, you bias it, but now if we use sequencing as a way, then you may see other microbes in there. But then it gets complicated because our microbiome is huge and sorting out.
Well, I guess a simple example of it would be all the secondary infections that people get when they have some condition such as HIV.
Exactly.
You've said, the HIV is often that's not the cause of death. The cause of death is some other thing that the immune system would have just shaken off, but because it was busy with HIV, now all of a sudden all these other normally innocuous threats are suddenly really serious.
Another one is secondary bacterial pneumonia after flu, right?
Yes,
exactly.
Right.
So that's why do those people get that? Everybody's colonized with these pneumococci, right? Right. Why do these individuals? I'll bet there's some mutation similar to the ones we're going to talk about today.
Right, and these are not mutually exclusive theories. You could have a combination of mutation that predisposes you to be co-infected. But this particular paper is a really cool genetic analysis that must have been a hell of a lot of work.
Yeah, it's quite a few, and that's why they have so many authors on here, I guess.
Yeah, I was going to say, so my wife is a clinical genetics fellow at Hopkins right now, and so she has all of these. Doesn't see actually. She sees more musculoskeletal diseases than immune diseases, but she has all of these families basically that they're trying to sequence and do SNP maps on and exome sequencing to identify novel variants that associate with the disease. So it gets very big, and it's actually interesting that there's some Saudi Arabian and Iranian kids in here or samples or doctors in here, because I'm sure the samples came from those families where there's a lot of consanguineous marriages and kids from where parents are related. So you can basically reveal these mutations, which normally you would not.
Yeah.
Yes, in fact, the first patient they talk about in this paper, patient 1, they mentioned that this was a consanguineous family.
Yep.
There you go. Yeah.
So
it's really interesting. I think, it's kind of like studying the Amish before, where it was more of a U.S. issue and people could get samples that easily. Now people can fly over, you get samples and you get blood or skin samples from people around the world. And if you have an infected kid or affected kid and a sibling, and then you have unaffected parents, you can now get a triplet or a quad to now start doing exome sequencing on to really start narrowing things down.
So the genes that they look at all have to do, as I said, with innate immune sensing, and these include the toll-like receptors, of which there are quite a few, and then the cytosolic receptors like RIG-I and MDA5, and these all are sensing foreign material, nucleic acids, or proteins, lipopolysaccharide. And the way these work is they recognize the foreign material, and then they initiate a signaling cascade, a series of protein phosphorylation events, which are very complicated, but eventually end up in the production of cytokines like interferon. And some of the intervening proteins in these cascades are the ones that they're looking at here, in particular the one called TWiV, which is in the title of this paper. This is an adapter in the pathway between TLR3 and the production of cytokines, and also TLR4. And it also seems to participate in the cytosolic pathways, the RIG-I and the MDA5. So we'll put a link to a picture in the show notes that you can see this as we're talking, because it does get a little bit complicated. So this first patient was from Saudi Arabia. The patient had encephalitis at 2 years of age. So the patient's okay now, is 3.5 years old. And the interesting thing here is has had no other severe infectious disease. So
this is not a kid who's just immunologically challenged or immunologically got problems.
Because we know there are mutations that cause susceptibility to many infections. You know, they eliminate B cells or T cells or whatever. So they sequence the genome of this individual. You get peripheral blood, you make DNA from it. Peripheral blood white cells, you make DNA and you can sequence it. And they sequence some of the genes that had been shown previously to confer susceptibility, some of the toll-like receptors and their adapter molecules, and they didn't find anything. But then they found a mutation in TWiV, all right, this adapter protein. It's involved in TLR3, 4, et cetera, signaling.
So it's another protein, but it's in the same pathway.
And this is a homozygous nonsense mutation, all right, at position 421. It causes an amino acid. Actually, it causes a termination codon.
Right, so you get a truncation.
That's what nonsense means. Homozygous means it's on both copies of the gene in this person, right? So that makes a short protein. And this is not in, they had 1,200 healthy controls that they sequenced, and they also looked at a database of sequences. They didn't find this mutation in anyone else. However, the parents of this child were both heterozygous for this mutation.
Right.
That's amazing, isn't it?
The same mutation too, which I thought was it. Yeah.
And in fact, they've got this pedigree. This paper, by the way, is open access, so people can read the whole thing if they want to. But in the first figure, they have this pedigree that shows. I guess they had 5 kids, and it's a textbook, almost a textbook genetics result. You've got the parents are both heterozygous, and you have one homozygous wild type kid, and then 3 kids who are heterozygous wild type and this mutation, and then 1 kid who's homozygous with this mutation on both copies of the gene.
Right.
And the parents did not have herpes encephalitis, so this is a recessive mutation. You have to be homozygous in order for it to be expressed.
Right.
So that's why they call it autosomal, because it's on an autosomal chromosome and it's recessive. So already you see a lot of work done here, thousands of sequences done as well. So this, they say, is probably a null allele because it truncates the protein.
Right. And importantly, in this figure also, they show you that the interacting domains in this protein where it interacts with the next protein downstream of it, those are all not expressed in this truncation either. So it's not if you had some of those domains where it was you took out the negative regulator, you could maybe overexpress this protein or overactive the protein. But this is truncating it very early, so you really lose all these interacting domains for the next the next handshake down the pathway.
Right.
Now, of course, this is only a handful of genes that they sequence, so you have to prove that these are involved, you have to do some more experiments, and we will get to those. But so far, it's a nice story, but it doesn't prove anything, right? Because this could have nothing to do with the encephalitis.
Right.
They also looked at a second patient who had encephalitis at 21 months. now 18 years old, no more infectious diseases. And again,
no unusually severe.
No unusually severe, yeah.
Nobody gets to be 18 with no other infectious diseases.
Yeah, it's true. They sequenced all these genes and found, again, in TWiV, a heterozygous substitution at 557. So this is now a missense mutation because it just changes the amino acid, goes from a serine to a leucine. And so this individual is heterozygous. So this must be if this is in fact involved in the disease susceptibility, it would be an autosomal recessive.
Dominant.
I was dominant.
Sorry, dominant.
So one copy is enough to screw things up.
Right.
And of this pedigree, one parent was heterozygous, and then a grandparent was also heterozygous for this mutation. But I don't think that either parent or grandparent had any herpes encephalitis. And this is another issue. Sometimes when you have these mutations, they are not completely penetrant, is the term. They don't. Even you may have it, but it may not express in you. Not everyone, for example, will get susceptibility to the disease.
Right.
So those are the two. There is a third patient, which we'll see later. But anyway, now the thing you can do is you can get skin cells from these individuals and culture them and do experiments.
Yep.
Right.
And they're pretty easy experiments. You put the cells in dishes and you add known ligands for these toll-like receptor pathways. And then you can measure the production of interferon. I mean, we do this in the lab with HeLa cells. It's really easy to do. And so they add, one classic thing you can add is double-stranded RNA. And then measure interferon or other cytokines. And they say,
right, because these pathways, that's one of their major triggers.
Right. So toll-like receptor 3 can be on the surface of cells, and if it is, it'll sense double-stranded RNA. You get this signaling pathway, which goes through TWiV, and you get interferons produced. So what they find is that there's a defect in interferon production.
Yep.
And that suggests that this TWiV mutation is the problem. They do a bunch of different cytokines, which we don't need to go into, but bottom line is it's defective. You can look at the intermediates. They actually look at the phosphorylation of transcription proteins. IRF3 is one of them. NF-kappa B is another one. And they showed there are defects in what you would normally see as well. So basically there is a problem in recognizing double-stranded RNA.
I would say from this still, it doesn't really tell you that these mutations are what's causing this.
That's right. That's right.
So this tells you they immortalize the fibroblasts, they make a cell line,
right.
But you still don't know that it's this one mutation that's really doing it. But you're getting there.
I don't know if you ever do, actually.
I think a good experiment, you kind of know what the phenotype of the protein is, but you could clone out the protein, take it by itself as a reporter into another cell line that lacks TWiV, and seeing if you can
have a defect,
see the defect there as well. But these are human genetics, guys. It's okay.
They do quite a bit.
No, they do. I'm just kidding.
So we should also say there's no protein in these cells. Or do they see a truncated protein or no? I seem to remember saying there's no protein, which suggests it's degraded. I think that's the case.
I don't know if I saw I don't think I saw a Western.
Yeah, these figures all have Westerns in them, but Western blot.
Well, they do actually. Yeah,
there's no
figure
protein.
2, they do a trunk. They show it. Yeah.
Yeah.
But the control is kind of wishy, so I don't know. That's not the best control.
So anyway, there's a defect in sensing, which might be associated with that. They also look at LPS, lipopolysaccharide, which is present in the in bacteria. It's a bacterial product known to trigger these toll-like receptors. And there's also a defect in that as well. And LPS is sensed by TLR4, which also interacts with TWiV. So that just tells you that TWiV is not working for both TLR3 and TLR4 pathways.
Right.
There's also some defect in the cytosolic sensing pathways, the RIG-I and the MDA5, which can use TWiV as well. It's not as severe because that's redundant in that case. So this has effects on multiple pathways.
I'm surprised they didn't see any bacterial issue with these kids never saw an issue with bacterial infections since you lose the TLR4 signaling also.
Yeah.
So what do you think that means?
There's obviously redundant.
I assume it's because I think it's because there's so much redundancy in the antibacterial defenses.
Must be, yeah.
Well, there is an antiviral too, but TWiV is one of the major players. It's not, MyD88 is also there as well to kind of protect you.
Well, and let's bear in mind these kids didn't seem to have problems with other viruses.
Yeah, it's very interesting that it's.
So it may just be that herpes encephalitis is very specifically needs this pathway to stop it.
Yeah, that's what they end up saying, is that probably when the virus gets in the brain, you need these interferon pathways to protect you.
Right.
But, you can't really conclude about other viruses because you don't know if they were ever challenged by others as well. You would have to challenge them. I mean, you could make a mouse with this mutation and go through a whole bunch of viruses and find out which ones are important or not.
Well, you can also do serum testing and see what they've been exposed to.
Yeah.
But yeah, you don't. This paper does not definitively prove that this is the pathway that's causing this disease, but it's starting to add up. You've got all these different components now, several different components that are all involved in this same antiviral pathway, have all now turned up in patients with herpes simplex encephalitis, and these mutations don't come up in other people. And
yeah.
Right.
So then they take these cells and they infect them with herpes virus, which is probably the experiment you like, Matt, right?
Right, exactly.
And they show with this mutation, the virus, kills the cells more extensively compared to controls, and you get poor responses of interferon upon infection, all consistent with this mutation conferring enhanced susceptibility to herpes, right? What they didn't do, which is interesting, they didn't put in a wild-type gene into these cells to complement it.
Oh, into the fibroblasts from the kids.
Kids, yeah.
Right.
They do clone the gene out, though. That's what the next, figure 5 or something is, where they take the gene out, express it in a cell that usually doesn't have TWiV, and then
show that it still has the defect. So what the defect they are seeing is due to that, right? So I guess that's okay. It's probably hard to put the gene into the fibroblasts anyway.
I don't think it's that hard, but it's okay. I think they probably cover themselves by doing the other experiment the other way.
Yeah, I think that experiment that they did is actually a little bit of a stronger case, because you're only moving this defective gene and you seem to move the defect with it.
Yeah, so they clone it out, they put it in cells, and there is still a defect. Essentially, you do the wild type gene, there's no defect in these null cells that Matt was talking about.
Yeah, a lot of times what they'll do is they'll get the same skin sample from the non-affected sibling, and then use that as your control, and show there's no effect there, but there is when you have the mutation, or when it's heterozygote versus homozygote. So that would have been nice to see. I'm guessing they didn't have the skin samples from the non-affected SIBs, but a lot of times they'll do that as well.
What I think would be cool to do at this point, now that the technology is available to do this, is to induce these to form pluripotent stem cells and grow neurons out of them.
And then infect them with herpes.
And then infect them with herpes and get an actual neuronal model of it.
Yeah, that would be cool.
Now that's a whole other paper, because that obviously requires a fair amount of work. But
so they conclude this mutation in this patient 1 is responsible for the TWiV deficiency and the abolition of TLR3 signaling and susceptibility to herpes. And I believe that. That's fine. Yeah. They move on to the next mutation. This is the autosomal dominant. Patient number 2. And this is an interesting journey, because they do a lot of stuff that doesn't work, basically. They just conclude that first of all, the fibroblasts don't have any aberrant responses. Right? They put poly-IC in, or double-stranded RNA, and they don't differ from wild-type controls.
Right.
And then they try overexpressing the mutant allele, and there's no difference. And they say, when you have too much protein, maybe it doesn't work. It's a lot of negative results, which are interesting in a journal article usually, and they're carefully documented. They finally end up putting this gene into cells using a lentivirus vector, which will maybe give you less levels of protein so it doesn't complicate it. And there they conclude that there is a defect in sensing in response to double-stranded RNA compared to wild-type cells. And they say these results suggest that the allele is hypomorphic, which I have to be honest, the first time I've encountered that term. Do you guys know about hypomorphic?
I do now.
Yeah, we have another allele in the lab we're working on that's hypomorphic.
Hypomorphic,
yeah.
It's a mutation that causes a partial loss of gene function. So you have a reduction of gene, protein, or RNA, but not complete.
Yeah, so a lot of genes you can't knock out in mice because they're lethal.
Yeah.
So there's mutations in some genes that have been identified that knock down the function near 90%. So it's enough to make the mouse alive, but you now have a reduction in that protein, in the activity of that protein. So
and that's a hypomorphic allele.
That's right. And there's hypermorph, which is the overactive phenotype. So if there was a hypermorph in this pathway, you would get, essentially constitutive activation of interferon signaling, which probably is as bad as down-regulating too.
Yeah.
So
now you may wonder why this mutation is a dominant mutation, right? And they don't really figure that out. But presumably the protein is misfolding, and that is interfering. And these proteins typically act with other proteins; they interact with them. So a misfolded protein may inhibit the function of the wild type protein that's present.
Right.
Right. So remember, this individual has one wild type gene and one mutant. And presumably, the mutant protein is interfering with the wild type, and that's why you have a phenotype. But it's not 100% abolished, so that explains their difficulties in vitro in figuring out what's going on. They also had a third patient, an Iranian boy who had encephalitis at 4.5 years of age, is now 7, again, not on any other unusual or severe infections. And they had a missense mutation in TWiV at 1875, which is further down in the protein than that original one. Again, so this is a change, leucine from a proline. And the father of this child has this mutation. But this is, again, it's a heterozygous mutation, one copy. And they found that the cells from this boy don't respond to poly-IC, the double-stranded RNA. Mm-hmm. But they couldn't complement it with the wild-type allele as they could previously.
I like that.
Why?
Well, it means that it's clearly still active at some level and be able to bind to its partner downstream, but it's not able to. It's able to basically inhibit. It's a dominant negative, so it's able to inhibit the activity of even a wild type TWiV when you put it back in.
Yeah, right.
It's great. Good. I mean, it's a great experiment. Humans are pretty good experimental models, aren't they?
Yeah. And they conclude we are unable to show that this particular mutation is deleterious with the assays available because it doesn't. Can't be complemented. Well, they said it was defective in sensing double-stranded RNA, so I think that's deleterious, right?
In vitro.
Yeah.
Yeah. You're probably backed up by the TLRs versus the RIG-I MD5 during a real infection.
They suggest that this patient has a mutation in another gene in the signaling pathway. So that's another problem. You may find this mutation, but it doesn't give you the phenotype you're looking for. So there may be something else. They have to keep sequencing. So this is the problem with these human studies because there are a lot of genes in there that you can look at. So this is cool. These are a couple of mutations in TWiV that affect susceptibility to herpes simplex encephalitis. So their hypothesis is that these life-threatening pediatric diseases are a result of single gene mutations that can be a problem, but they don't have 100% penetrance, which means that not everyone with them have the disease. And as I said, I think there are probably others as well, not just pediatric infections, probably adults too.
Sure.
It's just hard to sort out because our genome is so big.
Yeah, this happens to be a really good system for looking at this because the phenotype of the severe disease is so obvious.
Right.
And these are kids who come to medical attention for this specific condition, and then you've got these very nice, neat point mutations that have shown up in these studies. So it's a good system for looking at that. But a lot of the others are a lot more complex.
Yeah.
So if you were a new parent, would you want to have your newborn sequenced so you'd know if they have a TWiV mutation or not?
Yeah, a whole genome sequence for sure.
Even though you don't know what most of the mutations would do?
No, I still think if it were $1,000, I would definitely do it.
Yeah, I probably would too.
I'd do mine now for $1,000 just to see what I'm heading for,
As long as I knew my insurance company couldn't get a hold of it.
Yeah, right.
Well, that's that's a whole nother level.
I mean, that's an issue.
I mean, in my family, one child never gets sick and the other two get sick all the time. It'd be interesting to know we wouldn't be able to prove anything, but
Right.
it'd be nice conversational information, if anything. Anyway, these are great studies because it involves a lot of people, and the PI has to be a really good coordinator to get all this together to work. So I really admire it, and there's good science here as well.
Yeah.
So, and you can imagine more stuff is going down with this as well. So that's that one. Our second paper is a MBio paper called Local Response to Microneedle-Based Influenza Immunization in the Skin. And I thought this would be good to talk about because this is a very cool way of immunizing. So they use influenza here because, as everyone probably knows if you've listened to TWiV, the influenza vaccines aren't great, and we could use more immunogenic, long-lasting vaccines. So what they're pursuing here, and this is from the lab of Richard Compans. The first author is Del Pilar Martin. And these are microneedles. These are little patches, synthetic patches, smaller than, say, your thumb. And they have tiny needles on them, which just go through your epidermis.
Sort of medical Velcro.
Medical Velcro, exactly.
I like that. Very good.
And this has actually been studied for quite a while. They basically saturate these with vaccine. It's frozen. Then when you go to get your vaccine, it's put on your skin with a Band-Aid, basically. And you leave it there for a while, and the vaccine thaws. It gets absorbed into your skin. And it's apparently painless. I can't vouch for that because I've never had it, but I don't get much pain from the other ones either, so it doesn't bother me. But it's cool because it's painless. It's probably easy to distribute, and
wouldn't require skilled people to do it.
Yeah.
Nope.
Now,
you could shoot it out of a gun from a distance.
The skin is getting a lot of attention for immunization because it's full of dendritic cells and other sentinels, and it's a great immune organ. So, in fact, I didn't realize this, but a dermally administered flu vaccine has been approved.
Yes.
Not by these microneedles, but by a special kind of needle that just goes, right into the skin itself.
Yeah, I was actually I was at the flu vaccine summit last year in May, and Sanofi representative gave a talk about this new subcutaneous flu vaccine, and they've got that's a much simpler device. I shouldn't say that. It actually took an enormous amount of engineering effort to get it to work, but the idea is it's just a very, very short needle in this whole cartridge, and you put it on somebody's arm and just push a button, and it springs this little needle into the skin and injects exactly the right dose right under the skin. And again, the emphasis is immunogenicity because you get it into the skin and it's potentially more immunogenic there, and also easier delivery.
This looks like a complicated thing here, probably expensive to make.
Yes.
So intradermal inoculation is hard. If any of you have ever had a tuberculin test, you know that they have a small syringe, less than a mil capacity, 0.1 ml, and then the needle has to go intradermally. So they usually hold it horizontally on your forearm, and the nurse or whoever gives it to you has to be very skilled to do this. I remember watching once as she did it. She was she teased the skin and just put it right in between the layers of skin there, and you put a little tuberculin in, you see if you get a immune reaction. So that's how you would have to immunize intradermally. So these micro needle patches deliver it to the same place, but they're much easier because you just tape them on with a Band-Aid.
Right.
So I think this is a very cool technology, and as you will see from this paper, it looks like it works really well.
Yeah.
So this is not the first paper to try this. In fact, they have shown before that if you immunize mice with inactivated influenza vaccine using these skin patches, I think they put it on their ear,
right.
And you get very good immunity. You get protective immunity against the same strain. It lasts a long time, and you get good cellular responses as well. So this is a paper to characterize it, but I thought we would use it just to show this technology and how it works. So again, the skin is full of all these antigen-presenting cells, dendritic cells, which we talked about. And remember, if you remember from our DC TWiV, the dendritic cells are the sentinels. They are patrolling our bodies looking for foreign pathogens or microbes that don't belong there. And if they encounter one, they'll take it and process it and bring it back to the lymph node and present it to a T cell.
And they're very active in the skin because a lot of pathogens end up coming in through little breaks in the skin.
Right.
Yeah, the skin is full of them. So it turns out that the skin is better than the muscle for sticking in vaccines, right? Because the muscle is like, nobody's going to hurt us down here deeply in
the
your
skin's got us covered, literally.
The skin's got us covered, that's right. So they do this very interesting study in this paper to find out what's going on when they immunize. So the first thing they do is look at cytokine and chemokine expression. So the idea is that if you put an antigen in, you get a lot of cytokines and chemokines produced that are going to recruit cells in, including DCs, macrophages, and other sentinel cells. And that'll help get you a really good immune response. So they immunize their mice. They put these microneedle patches on them, and then they just take the skin and they measure cytokines, which is, again, pretty easy to do. And indeed, they get significant increases in a variety of different cytokines that are known to be chemotactic, in other words, attracting immune cells in. What I find...
It's important to do this sort of analysis because, obviously, as you said, they've previously found this vaccine seems to work pretty well in mice, but you'd really like to know how it works.
Yeah.
What's the mechanism?
Yeah, because if someone wants to license this, you have no idea how it works. It's not very good.
And we have the tools where we can actually look. So here they're doing the really detailed work of what exactly is going on when you stick this patch on. So you get the cytokines.
What I found interesting is if you just put the patch on, you get some cytokines.
Right.
Isn't that neat? So just a little damage to the skin initiates this inflammatory response. It's very mild, of course, because there's no antigen and it goes away. But
right, you get a much stronger response with the virus, but just the insult itself sets off the alarm.
So that's why you get a little scratch on your skin. It gets red and swollen. That's why, because you're making cytokines, and that causes inflammation and swelling.
Right.
It's really cool, Matt.
And I would say there's probably also, other pathogens in your skin you don't even know they're there. So even just breaking the skin surface, you're now allowing that to be picked up by DCs and presented. So,
yeah, of course we have a skin microbiome, and that's probably putting it in. And then the DCs say, these are our friends. We don't have to worry about them. And they just go back.
As long as they don't go any further.
So you get this very nice cytokine profile, all the right cytokines for recruiting immune cells. Then the next question is, if you use these microneedle patches, do the DCs go out from the immunized area? Do they migrate out? So they immunize, and then they excise the skin, and they put it in a dish, and they measure the cells exiting the skin piece in the medium, which they can do by just measuring the number of cells, and they can characterize them by flow cytometry, say what kind of them are. And so they say at 2 hours, half of the dendritic cells were influenza antigen positive. So they can actually say, are these not only DCs, but do they have flu antigens on their surfaces? Are they presenting flu antigens?
Right. So they detected it in the skin, and then the dendritic cells decided to run out and tell somebody.
Right.
Probably.
So they ran out. So this is good because that means they're migrating. Now, where they would migrate to is to the lymph nodes, but they weren't able to find any flu-carrying DCs in the lymph nodes within 3 days. And I don't know why. They don't even actually talk about that very much. This is what you would expect would happen, right?
Right.
But they don't.
Yeah, you should see them there.
Yeah, they don't. So that's good. The DCs are migrating out of the immunized area. That's what you would do. Now, one thing that they use here, which I also found very new, is they label their virus with a dye called Q-dot. Have either of you ever encountered Q-dot dyes? Matt, you might have.
Yeah, there's someone in our apartment who uses them for kind of NKT cell cancer therapy tracking. So they're very cool. It's amazingly bright, and you can basically label protein. You can label cells with them. They uptake. They bind to different antigens. If you can link them to whatever you want, and you can now use that as a way to follow where the antigen or the cell goes in a mouse or in a cell culture or wherever you want.
Yeah, so they're about the size of a protein. You can link them up to various antibodies or whatever. Yeah. And as you said, they're very bright, and you can make many different colors.
Right. So you can do all different. You can, I think, depending on the way however many colors your fax machine has.
Yeah. Right.
FACS, not FAX.
Yes.
For everyone who doesn't know what that means. Flow cytometer. You can really follow these things, and they're very stable. They dilute. I mean, if you label a cell, it dilutes out as the cell divides, so they're not great for long-term stability. But for this type of experiment, it's perfect.
Yeah, they have. I was looking at the website. These are made by Invitrogen, which is now Life Technologies, and the technology, nanocrystal technology, it was done at St. Petersburg in the former Soviet Union, also at Bell Labs, and Invitrogen licensed it. They're the sole licensees of this Q-dot technology. It seems, and the pictures they have there are just beautiful. They have one picture of a kidney section stained for various proteins. It's just bright and sharp, really nice.
Yeah.
So they label their influenza virus in this study with that. We'll put some links to that so you can check that out. It sounds expensive to me.
The Q-dots, they're not that bad.
Not too bad. No worse than, say, Alexa Floor type things.
Yeah, no, I mean they might be a little more, but they give you a lot more stability than, and you can't really do this with that kind of Alexa.
Yeah.
Some kind of modifying dye that way. So people are starting to use them a lot now. For people doing it for a lot of neural sections and things like that, where you can kind of follow it across a neuron and watch it move with a protein. So after you inject it in, so it works really well.
Yeah, and I think Invitrogen relies on selling volume.
Yep.
So they know you're going to come to them for more of these after you use them.
Yeah, I'm trying to think. A couple other companies have versions of this. I can't remember which ones it is, but I think all of the major the biology companies are now starting their own version to kind of, maximize everyone else or spread out everyone else's use,
sure,
so they can get some of the money.
Yeah, and the initial patents on this are expired, I'm sure.
So the next thing they do is look at where this antigen is in the skin. So, you're putting a microneedle patch on. Is the antigen staying there for a while? Does it go away? So they use first ovalbumin, just a protein, a common protein, and put it on the needles and say, how long is it retained in the skin? And they image the skin for up to a week after immunization, and they look at where the protein is. And then what they find is that you get a lot of protein deposited at the site of immunization, and it stays there for a long time, up to 5 days afterwards. And that they compared it to, say, an intramuscular injection, and it lasts much longer than in an IM. So this is good because you want your antigen to stay around, especially in the skin, if that's where the priming is going on. 2.5-fold more antigen at the site of microneedle vaccination compared to intramuscular vaccination. So that may be part of why it works so much better in the skin. Then they confirm this with virus. They use Q-dot labeled viruses, and they say you get similar results with influenza viruses. So this is called the depot effect. When you put antigen somewhere, it stays there for longer than you would expect. And that's usually good to stimulate immune responses. It's also how adjuvants work. So when you mix an antigen with an adjuvant like Frieman or aluminum hydroxide, you make depots, antigen depots, and this is thought to contribute to the improved antigenicity. Of course, adjuvants also increase inflammation, that is, the recruitment of cells via cytokine production and so forth. And that's another reason why you get a good immune response. So these microneedles, they give you good cytokines, and the cells that are attracted there, the dendritic cells, migrate, and the antigen lasts a long time. So these are all good, all components of probably why that this method of immunization is so good.
Yeah, and these are non-adjuvanted antigens.
That's right.
Right?
Yeah, they're
so
non-adjuvanted.
I wonder. So you don't have to worry about any of the people saying that there's an adjuvant effect. But I wonder if even if you adjuvanted them and you put in some of the other normal adjuvants that people use in vaccines, if you would, or even pro-inflammatory cytokines for that matter, that you could actually boost this response.
Well, the microneedles themselves have a little bit of an adjuvant effect. You get a little bit of a response when you just slap the thing on with no vaccine on it. So you are getting a little bit of inflammation that's just stimulated by the patch itself. But yeah, it would be an interesting experiment to try doing an adjuvanted vaccine the same way and see what effect you get.
Yep. So this is very promising. I like this idea. I think the results look good, and I think I would say that this is going to probably make it into people at some point.
Yeah.
Now, it's interesting. Now, you may be thinking, no, wait a minute. I'm putting a patch on my skin, and it's protecting my respiratory tract against flu. And because the antibodies, once they're induced in your lymph nodes, they go everywhere.
It's no stranger than sticking a needle in the muscle, and that protects your lungs.
Right. So I like this technology. I thought we should introduce it to everyone.
Absolutely.
And in this paper, they don't do any challenge studies in the mice, but they did this in another paper. So this paper was more looking at how it works.
Yeah, and the challenges look good.
And the challenge, in the previous paper where they just did challenge, but didn't look at any kind of, what was going on immune wise.
Yeah.
They work really well. So at least in mice. Again, all this is in mice.
So they say here that the immunization produces robust serum antibody responses and cellular responses capable of conferring protection against challenge at least as effectively as most conventional routes. Importantly, antibody titers remain elevated in the microneedle immunized group even 6 months after vaccination and correlated with inhibition of viral replication and cellular responses. So they get good responses in long term, which may be really a plus here because, as we know, the injected flu vaccines aren't very durable, especially in older people.
Right.
Yeah. They also don't. The other thing, which I'm guessing is the next step, is to make a combination vaccine. So in here, they're just doing one antigen for the flu vaccine. So in all of our flu vaccines we get injected, it's a triple vaccine. So you have to be able to show that it works for. You can spread out the antigen load. And do it that way. But all looks good. I mean, I'm sure we're going to see these soon, I would imagine. Soon being, 10 years? I don't know.
10 years. Well, we have to do it in ferrets next, right? See if it protects against aerosol.
That's right.
I wasn't even going to, I thought we'd be a ferret-free TWiV. I guess not.
No, sorry. The other thing is, I wonder, so everyone is working on these broadly neutralizing antigens now for flu, right?
Right.
I just wonder if it would be suitable for this kind of delivery and whether being recognized by DCs in the skin would give you a different kind of antibody response, which would even help these broadly neutralizing antibodies in some way.
That would kind of be the dream, wouldn't it? You have a patch that you slap on your kid's arm, and a week later you take it off, and for the rest of their life they'll never get the flu.
That's great. If it worked, yeah.
Yeah.
All right, let us do a couple of emails. The first one is from Mark. He is writing with reference to the poultry staggerers from the TWiV 174. We had an email about that. He says, Marek's disease would be worthy of consideration as a cause of the staggering, but there are a number of other possible etiologies, ryegrass staggerers, and dietary deficiencies.
I've staggered because of rye before, but I don't think that's the same thing.
Ryegrass, yeah. So Marek's is a disease of chickens caused by a herpes virus. And he's suggesting that they could stagger and be mistaken for it, although I don't know if chickens in Texas stagger because of this. But anyway.
I still think it's heat stroke, but this does look like a possibility.
Yeah. All right, the next one is from Chris, who writes, Vince, Rich, Allen, and Dixon, this is a long overdue email to express my gratitude to you all. I am an assistant professor in virology at UT Austin and have been hooked on your show since I became a new dad 18 months ago. I'll get back to that later. I would like to briefly highlight some of the ways TWiV has helped me. First, as an educator, I teach an upper-level undergraduate course on animal virology. There is no doubt that I am a better teacher because of TWiV. My breadth of knowledge of virology is greatly expanded as a result of my weekly TWiV fix. As a result of this, the students think I am smarter than I actually am. Your show has inspired an ongoing experiment in my class where, as a teaching tool, we incorporate a student-selected current event relevant to virology. Topics typically come from the lay press and are scientifically dissected at the beginning of every class. This appears to be a big hit with the students, especially those who came into the course as a forced requirement as part of their degree plan. Rightfully so, nothing piques a student's interest more than the relevance and timeliness of the topic. In addition, my students are offered as extra credit the option of summarizing an episode of TWiV. Typically, 60 of my 90 students opt to participate in this exercise, and their response has been very positive. Several like it so much that they end up listening to many episodes. That's cool.
Excellent.
Very good.
Second, your show has helped me to be a better scientist. In addition to increasing my knowledge of tangential fields and new techniques, there have been several times where a new line of experimentation for my lab has emerged from TWiV. One example that comes to mind came from something Rich said, I believe, regarding consideration of temperature in experiments. I believe this was with regard to TS mutants. This dialogue on your show made us consider the simple idea that temperature could account for the lack of an infectious tissue culture model for an upper respiratory virus that we are studying. We don't know yet if this is the answer to our problems, as we are still waiting for the new cooler temperature incubator to arrive. Nonetheless, this is a reasonable hypothesis to test, and this entire line of experimentation was inspired by TWiV.
Let me just jump in with a pointer here.
I was going to invite you to.
Yes. For my thesis research, I needed to incubate my samples at 25 degrees C, which is allegedly room temperature. But of course, the rooms in the building where I worked fluctuated from, 15 to 30 on a typical heat cycle. So what I did was I just took a conventional incubator that we had, a conventional cell culture incubator, put it in the cold room, and I found that gave me, I could get it to 25 degrees, and of course the cold room will handle keeping it cool. And if you need it more accurate than that, putting a computer fan in it to circulate the air was extremely effective. I ended up with an incubator that was accurate to plus or minus 0.5 degrees C. And that was with an NIST certified thermometer. I mean, I went the whole 9 yards on that. But the thermometer was, $50. An incubator, I looked at incubators that would actually have maintained 25 degrees, sitting in the room, and they were thousands and thousands of dollars. So if your incubator hasn't arrived yet, just pop one of the ones you have into the cold room and go for it.
I could vouch that it did work.
Yeah.
All right, continuing, as also in regards to how your show helps scientists, I must mention the TWiV bump. I believe that at least Alan is a Colbert Report fan based on his reference to truthiness on TWiV.
That's Colbert Report.
Yes.
As you may know, Stephen Colbert claims that artists who have their work mentioned on his show benefit in terms of recognition, sales, prestige, etc. He refers to this as the Colbert bump. I think it's obvious that this is also true for scientists whose work is mentioned on TWiV. In TWiV 174, you profiled a paper from my lab. Mere hours after that episode was released, I was contacted by 2 different colleagues offering me their congratulations and envy. You should know that it is a career goal of many of us younger virologists to get the TWiV bump. That's just great.
It's very good.
TWiV bump. Did you get a TWiV bump, Matt?
I have, yeah. We have. I can't tell you how many people have emailed me about either wanting to go to grad school here because they heard me on talking about TWiV in our program, or wanted to talk about bats. Or I actually gave a talk at our neighboring institution, Johns Hopkins, for their microbiology or in virology class, and it was the week after I was on TWiV last time.
Nice.
And so the kids had heard me on Monday morning and listened to it for hour and a half of me blabbing, and then had to listen to me for another hour and a half while I talked about SARS. So they had 3 hours of me in a week, which may be the downside of being on TWiV the week you're in a lecture. But no, I have to say, we spread far and wide. You'd be amazed at how many people are really listening.
That's great. I love it. Finally, it is also true that you make parenthood easier. When I was a brand new dad and got to spend hours and hours with my son Sam, TWiV served a valuable role. Sam and I would go on long walks, and TWiV made the time fly by while appealing to my sense of productivity. So not only is your show helping research professors and high school students alike, arguably it serves a positive role for some infants too. I think you do that, Matt, with your child, right?
Absolutely. Well, my daughter's, I have a 2.5-year-old daughter, and while she's listening to Sesame Street videos on the iPad on the way to daycare in the morning, I am, a couple morning or two a week, I listen to TWiV. Keeps me sane instead of listening to Elmo for 40 minutes on my drive in.
Maybe she's absorbing some virology.
I hope so.
Someday you can ask her a question.
Alan thinks Elmo should get a flu shot.
In summary, I'm grateful to you all, knowing firsthand the demands on a faculty member's time and imagining it's similar being a freelance writer-reporter. I don't know how you find the time to do this. It is a wonderful service to science and science education. As an NSF-funded researcher, I hope the NSF sees the value in what you are doing and ends up supporting TWiV. I hope you keep up the TWiVing for a very long time. So Chris's paper, of course, we did last week. It's the microRNAs in the bovine leukemia virus.
Right.
That we talked about. Chris is an assistant professor in Austin, University of Texas. And he says maybe we'll try and get the TWiV team there. So maybe we will go.
Sure.
Alan, can you do the next one?
Sure. April writes, "Hi, Vincent et al. Great show this week. On the note of coffee makers, this one beats them all for taste, in my opinion. Plus, it's made in the Berkshires." And she links to the Chemex coffee maker, which I think it looks kind of like an Erlenmeyer flask with a funnel.
Yeah. I've seen those. Yeah.
Kind of a neat little concept, but it's got this nice little wood piece. Okay, cool. I just have an ordinary. I don't even know what it is. It's a Mr. Coffee or type thing or something, but it has the clock on it, so it comes on automatically.
Yeah, no clock on this one.
No.
no. This looks very old school, but it looks cool.
This is going back to the. I'm sure it's Brooklyn-made artisan wood carved.
Well, it's Berkshires, so that's up near me.
Yeah. There you go.
April is at the Wadsworth Center up in Albany.
Yep.
She says she likes to hear it on her commute from the Berkshires to Albany. Well, that's a nice commute. Nice to live in the Berkshires. The next one is from Aisha, who sends 3 links to articles about the Elsevier legislation to stop the open access. She writes, "I'm trying to form an opinion on the subject. I'd love to hear what you have to say on the matter and why Elsevier are lobbying for the bill. What will it mean? I'm a bit unclear." So we talked about it on 173. Mm-hmm. So check that out. Okay.
And they have now, Elsevier has now pulled out its support for this act.
Right.
Exactly.
Because they didn't want to not have their researchers reviewing papers for them.
Right. Matt, do you want to read one? Do you have the mail in front of you?
Sure. Yeah, I do. So Tessa writes, "Hello, TWiV. I'm rounding up my 4th year in my thesis lab, and currently the stacks of half-read, highlighted, triple-printed papers are starting to take over all my usable space on my desk."
Been there, done that.
Yes. It's a good 4th year. That's about right. It starts getting to where you can't sit down anymore. I think the only way I can keep sane in graduate school is by keeping myself organized and regular doses of therapeutic humor from PhD comics. Technology is getting so sophisticated. Do you know of an online service where a person can electronically accumulate and organize all of these PDFs, be able to search for keywords, highlight, add notes, etc., and be accessible from any computer, lab, or home? I'm super excited to attend a live recording of TWiV this summer at ASV's annual meeting, hosted by the University of Wisconsin-Madison. Thanks to you guys, I'm proudly embraced by virology geekness, sharing the same sentiments as all of your viewers. Keep up the good work. Tessa, who's a PhD candidate at Thomas Jefferson University in Philly.
Cool. Yeah, embrace your geekiness for sure.
Very good.
I don't know if it does everything you want, but I know Mendeley and Cite You Like are places where you can archive PDFs and references. I don't know if you guys have used any of those.
I have not used either. I've looked at both of those. I ended up settling on Zotero, which is another one. The thing I like about it is there's a convenient Firefox plugin that works very well for my workflow. In fact, I've been using it on this show. I've got my notes for the papers right in Zotero, and I've got the papers linked. And you, Tessa, you may want to think twice about saving them all as PDFs. I find it's actually useful if you have just links to the articles. If they're full text online, that's a particularly good way to do it. But up to you. And all those tools will probably do more or less what you want.
Do you have any solution, Matt, that you like?
Yeah, the other one that I have now is Papers. Yeah. It's just called Papers, and it's a desktop app, and they have an iPhone app and a iPad app, and you can download and save the PDF in there, and then it syncs between all the different interfaces. So you can go in there, you can highlight, you can do PubMed searches, download the PDF, saves everything in there. I'm still getting used to it. I use it a little bit here and there, but it seems to work pretty well.
And you can have one database and sync
you sync between everything, right? Which is really nice.
You use Dropbox or something like that to store the database, or
no? It syncs it all. I mean, it syncs through Papers.
I see.
So Papers does it through the cloud, and it syncs it between
okay.
all of the things. So it works really well. Yeah, the other way is Dropbox, which you can set it up so it syncs from whatever service you want, and just drop the PDFs in. And, I was actually wondering on the last TWiV, maybe it was two ago, you said that you were looking at different annotation programs. Have you decided on one yet?
Yeah,
I've been playing around too, and I haven't found what I like.
That's going to be my pick.
Okay.
I'll tell you. So that's what we think, Tessa. And if anyone else has solutions, send them in. We'd like to hear them.
Yeah, and there's competition among these, so shop around, and you can definitely. This is a problem that people are waiting to solve for you.
Yeah, actually, it's funny that this came up this week. My brother and I have a small idea to be able to do this for academics. So maybe we're looking for programmers now, but we have a little side idea that may come to fruition. But for doing almost exactly this. So
cool.
We'll see. Maybe
do Kickstarter.
I'll let you know when it. Yeah, okay, that's true. We should do Kickstarter. I'll let you know when it comes through.
Alan, would you take the last one?
Sure. Catherine writes, Dear regular TWiVsters and guests, I listened to your last episode, number 168, while chilling out in the hospital. What better place to learn about viruses? I hope that worked out okay. I enjoyed the pick of Dr. Racaniello. I agree with the point of view of the teacher. As an ESL teacher myself, I can stand on my head and do backflips. The ones who want to be learning English do their homework and participate in class. The ones forced to there just do the opposite or worse. They disrupt class or demand, not ask politely to play games. Part of my teaching philosophy is to get the kids to learn without them knowing they were learning. I remember fondly watching Mr. Wizard's World growing up. I didn't realize how much I learned through that show and how it helped me through elementary school science. Interestingly, if you Google the name of the blog post, what pick was this of yours, Vincent?
So this was an article by I. Cringely.
Right.
How technology is not going to help you teach, like iPads and so forth, because if the kids don't want to learn, they're not going to learn.
Right.
That's
right.
about.
Okay. So interestingly, if you Google the name of the blog post, you get an article about a South Korean, where I'm located, pilot project using robots to teach English. The picture shown is not a typical elementary class where there are 30 to 40 students, and the conclusions made at the end are a direct shot at the foreign teachers. I should explain the educational philosophy of South Korea. Kids go to the public school in the morning and then longer as they reach middle and high school, where they study the basic subjects. English education starts in the 3rd grade. Many then go to private academies for lessons in specific subjects. Those 3rd graders who have been studying English since approximately the age of 4 are light years ahead of their peers who didn't go to English preschools. There's no mandatory kindergarten. While a foreign teacher has some kids learning basic phonics, she has others not paying attention because they're learning more grammar than I know. In fact, the government is planning on phasing out native-speaking teachers by 2015. They say the students are more comfortable learning from an English-speaking Korean teacher. But the fact is, they can get away with speaking Korean in that model, but not with a native English teacher. I teach at one of the private schools where I have students from 6 to 14. At least they're roughly grouped by ability. I do not allow students to use their cell phone dictionaries or portable dictionaries in my classroom. I find the students use it as a crutch and don't learn to use context clues to find meaning. I have my phone, and if we as a class can't figure it out, then I'll look it up and read the Korean word. Now for my virus question. I am working back through the archives and just listened to the one on virus structure. Please correct my interpretation if I get it wrong. We start out with the simplest form, such as TMV, where the virome is simply wrapped in protein. Genome is simply wrapped in protein. This only works primarily in plants. When we get to non-plant hosts, you can have a similar structure enclosed in a lipid protein. Am I understanding that this comes from the host cell as the virus replicates and breaks out of the cell? I think you mean enclosed in a lipid envelope?
Yeah, that's right.
Right. So it's just a lipid envelope around the proteins.
And the specific word for getting out of the cell is budding.
Right. Yeah. On another level of complexity, up is the envelope. That actually is the envelope, the lipid layer. Here there are 2 layers of lipids with protein in the middle. Yeah, I think you mean lipid bilayer? Is that what you
Yeah,
mean?
there's no protein in the middle.
There's no protein in the middle. When we say lipid bilayer, we mean that's just the way lipid molecules organize into a membrane. They have the part of the molecule sticking out toward the water side, the aqueous side, and then the tail of it is sticking in toward the middle. And you have two of those tail to tail making up a bilayer. Mm-hmm. So lipid membranes are made up of a bilayer, but there's nothing. It's not that they have a layer of protein in between. What is the advantage of that? Does it make the virus more resistant to the host's immune defenses?
So there are no. She's thinking of these two levels of lipid bilayer, but it's all the same. It's a lipid bilayer, and it's there to protect the genome, basically.
Right.
Why plants like TMV, plant viruses like TMV, don't have it is not clear. But we don't see those naked genomes like TMV in animal viruses, so there must be a reason for that. It's probably protective. And also you can put receptor binding proteins into a lipid envelope and allow it to attach to the cell.
Yeah. The most complex is the protein shell. Describing it as such makes me assume that it is more rigid. Well, the envelope viruses generally also have a protein shell underneath the envelope. So there's the protein capsid, and then around that is the lipid bilayer ensheathing it. So that's why they're called enveloped, because you have, whereas the ones that don't have the envelope are called naked. So you have naked viruses that are just the protein capsid. Then you have enveloped viruses where you take a naked virus and you put it inside a lipid envelope.
Right.
And then she asks, why is icosahedral the only type of symmetry? Wouldn't a cube be as simple, 6 squares versus 20 triangles?
Yeah, good question.
Yeah.
It's actually an icosahedron gives you maximum internal volume with minimum number of components.
Right.
And a cube just wouldn't do that.
That was the Buckminster-Fuller dome.
Yeah.
Which unfortunately they all leak, but viruses
viruses don't leak.
Viruses have it easier. They're enclosing this genome, which is different.
So that's true. It's the geometry works, and viruses adapted it or evolved to use it. It's interesting.
Yeah, the ideal shape would actually be a sphere, but you can't make a sphere out of
individual components.
A finite number of individual components. So the best compromise turns out to be a two-sided figure.
In fact, most icosahedral particles look spherical. Anyway, so it's the best approximation of a sphere, probably, right?
Right. So then she asked, does what exists in nature give the most bang for the virus's buck, so to say? And yes. That's exactly how this works out, because viruses that adopted a different shape had an evolutionary disadvantage. So the one that was able to build its maximum volume capsid with the least energy, that's the one that won the evolutionary race. It's the most effective with the fewest number of parts. Or is there some underlying factor in the tertiary, quaternary form of the proteins themselves? No, it's geometry, and the viruses arrived at that solution because that's the one that works. And then she finishes up, I again want to thank you for addressing my opinions and, basic questions. If TWiV had existed 20 years ago, I might be a virologist. I do hope some of my students go on to be scientists, as they have said when we talk about jobs and what we want to be when we grow up. I wish I had an answer for that last question myself.
It'd be interesting to see in 10 or 20 years how many virologists went into the field because of TWiV.
All of them.
Yeah. Well, if I'm around, we'll find out.
See, there you go. You need your TWiV. You need a TWiV head. What's a TWiV follower called?
It's a good question. I don't know. TWivite? TWivome?
Vivian?
I don't know. But if I'm around in 20 years, I will be TWiving, and we will ask people, if you're a virologist, did you get your start with TWiV? It would be great, because I don't think there's anything better you could do is to get people to go into a field by inspiring them, right?
Yep.
Right.
All right, let's pick some picks of the week here, and we will start with Matt.
Sure. So I was given a book when I graduated from my PhD program called Phage and the Origins of Molecular Biology, and this was given by my PhD boss gave this to me, and it's not new. I'll tell you that. It is. I'm trying to look. It was published in 1966, but you can still get it on Amazon. And it is basically a bunch of essays about the phage and kind of the origin of all of phage genetics and phage biology, and really the beginnings of all of the molecular techniques and molecular things that we do in the lab now. And it was initially made as a kind of an homage to Max Delbruck, where most of the people that are in this book, including Watson and let's see, Delbruck,
Sal Luria.
Yeah, Luria,
Gunther
Hershey,
Stent. Yeah, it's great.
Seymour Benzer, and all these guys were around the same time. Frank, Messelson and Stahl, Watson, they all go through their kind of original experiments and how they got to where they did, and talk about the old phage groups. And I read, it's on my desk actually, right on the bookshelf above my desk, and I look at it all the time because I always think that how fun it would be to be kind of the origins of mammalian virology and molecular virology, to see what it was really like, and to get to these. I mean, it really was a much smaller group than if you go to ASV and one of our big virology meetings, and there's thousands and thousands of people there. So
it's a great book.
I think it's really good. So you can definitely pick it up at at Amazon, and I'm sure most of the university book or library should have it, I would imagine. So could definitely take a look.
I have a copy from when I was a student. It no longer has a back. It's just individual pages. I have looked through it so much. I just love it. I think it's
Isn't it great?
Fabulous book.
This one and the other one is the Eighth Day of Creation.
Yes, I heard someone sent me that, in fact.
Which is really good too. I remember reading that when I was studying for my qualifying exam at Hopkins for grad school, and it's just I think if you are sitting in lab for your PhD, or even outside, not doing a PhD or not in science, but really want to read about kind of the origins of everything that we've talked about here, and everything that started with all the molecular biology and discovery of DNA, and how all of it really works together. It's really great to see the beginnings before you think of all the details and how kind of deep we are getting right now.
It's a good one. Check it out.
Cool.
Alan, what do you have?
Well, my pick is the Digital Imaging Institute. I just found these guys recently for a story that I was working on, and this is it was founded by a fellow who was trained as a fine art photographer and commercial photographer, and then he got into scientific imaging, and he's been doing imaging for ophthalmology for a number of years now. But he realized fairly early on, well, about 4 years ago, I guess, he really got the impetus to put this together. He realized there's a whole lot of training for things like Photoshop out there, but it's all aimed at people who are putting together advertising campaigns or magazines or other, creative photography types of things. But scientists use this stuff all the time, and there isn't training that's specifically tailored to them. So, my experience of using Photoshop in the lab was you just kind of figured it out. And now with the software getting more and more complex, and a lot of the image analysis that people are doing getting more and more complex, this is actually kind of a problem. Because if you're going to pull quantitative data out of your image, then it really matters exactly how far you moved all those sliders in Photoshop. So they've got this Digital Imaging Institute has now gotten a bunch of people who do scientific imaging and some people who do creative imaging, and they offer classes for Photoshop, and they also have online forums for people to talk about various issues that come up in digital imaging and specifically for science. So I think that may be useful to some listeners.
Yeah, I need a Photoshop course. I do what you said. You just learn what you need to do to get the figures made.
Exactly. And if you're just doing something that's okay, here's a gel, and all I'm going to do is crop it, and that's it, and that's fine. But as soon as you start adjusting levels, or you're going to do something else, well, then how much have I changed the data? Am I going to analyze the density of these spots, and how do I do that without skewing the results? So
cool, great,
very good,
thank you. All right, my picks are 2 iPad apps. So as Matt said a couple of weeks ago, I mentioned I was going through apps trying to find ones that would be good for reading PDFs and annotating them. And Alan, you had suggested using iBooks, so I checked that out, and it turns out you can't highlight in iBooks. You can read them and put them on your shelf and all, and that's cool, but you can't highlight them. So
yeah, I'm doing it on a phone, so I don't really For me, it's just to kind of glance at the abstract.
So a student in our lab who's in an adjoining lab, Michael, mentioned, check out Goodreader. He had listened to the podcast, and I realized I had Goodreader on my iPad, but hadn't really used it much. So Goodreader is very nice. You can read PDFs. You can highlight them very easily. And the neat thing is you can have them in your Dropbox and sync it. So if you make some annotations on your PDF, it syncs with your Dropbox automatically. So that's what I wanted, and that's how I've now been reading a lot of the papers for TWiV and my other podcasts. So Goodreader, I think it's about $9.99. I looked at a lot of others. I think this is the best one so far. It's not perfect, but I would use that. And then if you want to take notes, someone else asked me, what do I do for taking notes? Notability is really nice for that. If you get a stylus like the Box Wave stylus, which I picked a couple of weeks ago, you can take notes and Notability. You can write, you can type, you can add images. You could take a photograph, for example, if you're somewhere and you want to annotate the photograph. And it has a very nice interface. One of the issues about writing on an iPad with a stylus is when you put your palm down on the screen, it screws up the capacitance. So there are a lot of solutions for that, and none of them are great. But this one, it has a little digital slider that you move up, and you put your wrist on the slider, and as you write, you move it down. And I found that worked pretty well. You can get very nice writing, and so I like it. It's about $2, I think.
That's good.
So, yeah, go ahead.
No, I was going to say, I've been looking for the same thing, too, so I'll have to try it.
Yeah, so Notability doesn't do the PDFs, but it does a really good job at taking notes. If you want to go to a seminar, and take notes and whatever, you can even add recordings, I think, as well. So Notability and Goodreader.
Cool.
Very good.
We also have a listener pick of the week. This is from Jane, who writes, just a quick email to say how wonderful it was to meet you yesterday in Amsterdam. I have been an avid TWiV listener since my first TWiV experience in 2009 and look forward to many more podcasts. If you recall, I mentioned to you a book I thought you and your readers might enjoy, Smoking Ears and Screaming Teeth by Trevor Norton. Professor Norton is a professor emeritus at Liverpool University, UK, having retired in 25 from the chair of marine biology. The book is described as a hilarious celebration of the great eccentrics who have performed dangerous experiments on themselves for the benefit of humankind. I regard it as a witty and informative romp through the history of scientific discovery and the self-experimentation practiced by the bravest. I hope you enjoy your upcoming visit to Dublin, and congratulations on yet another award. With warm regards, Jane, who I met in Utrecht last week at the meeting I was at.
Very good.
So Jane is actually from Ireland and is working in Amsterdam. And so she came up and she said, I'm a TWiV listener. I have a pick for you. So there you go. Thank you, Jane.
None of these experiments probably
fun.
Publishable now.
Yes, that's right.
If you wanted to do that in lab now, there's no way you could get that through.
Yeah, sure. But people used to do it all the time, right? All right, that'll do it for TWiV 175. You can find us, of course, on iTunes, at the Zune Marketplace, and at twiv.tv. We have an app at microworld.org that you can use to stream your episodes to your iPhone or Android device. And, of course, do send us your questions and comments to TWiV at twiv.tv. Matt Frieman, thanks for joining us today.
No problem. I'll step in any time for Rich. I'll do my best Rich Condit voice.
Great. I appreciate it. Matt is at the University of Maryland in Baltimore, where it's really warm now.
Yes.
Great.
It's
to have you again.
quite nice. All the flowers are blooming.
Excellent. Alan Dove is at alandove.com. Thank you, Alan.
Always a pleasure.
Good to have you. I'm glad you could make it. You were almost not going to make it at the last minute.
Yes.
But we are always glad to have you.
Something came up, but then it went away.
Came up and went down. Very
Yes.
good. I'm Vincent Racaniello, and you can find me at virology.ws.
Vincent, does anyone ever write you to do the tagline at the end, like they do for Radiolab?
Oh, that's what they do there?
They have the part of the intro thing is people call in and they read all the legal stuff at the end and who sponsors and stuff.
No.
No one's ever tried it? Oh, that's funny.
No, that's a good idea, though. I think it would be fun to have people do it, yeah.
You have to splice in at the end, though. That's the only problem.
Yeah, so another TWiV is viral type thing? Or
Right, and if someone else would have to say it, every week somebody could say the last part.
If anybody wants to do taglines, record them and send them in.
There you go.
Right?
I think people should do it.
You could do it. Why don't you do it right now?
Should I do my Rich Conant voice?
Do your own voice. Yeah, good voice.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is Viral.